Cadet, thanks for beaming up from the Academy. I'm glad you're here. Welcome to the Nova Science Station. Today we need your help with a particularly interesting occurrence happening throughout the galaxy. First, I'm going to turn your attention to the screen. You may remember from some of your courses here at the Academy that pathogens cause diseases in a variety of ways. One of the ways that we measure how these pathogens cause diseases is how many it takes for an infection and how many it takes to be lethal. So the median infectious dose, or the ID50, is the number of pathogens with which half the people exposed to that number will become ill and half will not. This is the same for the median lethal dose. Half the number of people exposed to that dosage will die, half the number of people exposed to that dosage will not. You may have heard of LD50s from some other things such as environmental toxins. In this case, the example graph that you're looking at shows that it takes 10 to the 4 pathogenic agents for 50% mortality to occur. Sometimes diseases change, and by knowing the median infectious dose and the median lethal dose, we can see how these changes are occurring. The virulence of a disease is the ability to cause disease, the ease with which that pathogen causes a disease, and or the severity of which that pathogen causes a disease. So what we're going to do is take a moment to review some of the factors that can cause an increase in virulence. An increase in virulence may be visualized on a graph like this by a change in the ID50 or the LD50. When we look at the change in virulence, we're typically looking at a number of virulence factors. A big one is the adhesion molecules. If you remember from your pathogenetic studies, a pathogen must first adhere before it can infect. Any mutation in these adherence molecules that make them either A, more strongly bound, or B, able to bind to new areas of the host cell will increase the virulence of that pathogen. Additionally, pathogens can produce exoenzymes, or enzymes that they excrete, that will then cause things to happen in the host. From ancient Earth, there was an example of a bacteria called Helicobacter pylori. This particular bacteria produced urease, a type of enzyme that would break down the mucin layer that protects stomach cells from stomach acid. By breaking down that layer, it allowed Helicobacter pylori to move into the stomach epithelial cells. The ability to produce urease is a virulence factor. Additionally, some pathogens can produce toxins. Endotoxins are typically found in the membrane, and exotoxins are typically created in the cytosol and excreted. Regardless, the presence of these toxins may increase the ability to cause disease, or it may allow for the disease to be more lethal. There are certain types of Streptococcus bacteria, for example, that produce a exotoxin that increases the virulence factor that increases its virulence by 10. So it's 10 times more infectious. Additionally, anything that causes an evasion of the immune system is a virulence factor. For example, some bacteria have capsules. These capsules are not strictly bound to the cell and can be abandoned if necessary. Let's imagine a scenario where a capsulated bacteria has been identified by your immune system. By slipping out of this capsule, the bacteria emerges unscathed and can continue infecting. When we look at how these things happen, we come straight down to evolution. The random changes in these products that will cause increases in virulence. We have two terms for this. One is antigenic drift. Antigenic drift is literally evolution, a random mutation that impacted the antigen, or the small amino acid sequence that your immune cells recognize, so that it is no longer recognizable by your immune system. If this occurs by random mutation, it is called antigenic drift. Another manner that this can occur in, and this is very common in viruses, especially multi-strand genome viruses. To use an ancient Earth example, influenza. Influenza has 8 RNA strands. Should a human cell get infected by one virus particle, we'll call it virus A, there are 8 virus A RNA strands present. When that same cell gets infected by virus B, there are now 8 virus B RNA strands present. During viral replication, 8 strands are going to be put into virus C. These strands are often a reshuffling from the 16, 8 from A and 8 from B, so that virus C has a completely different genome than either of the ones that came before. This often results in changes to the antigens that your immune system recognized. It also can change the way that the virus is able to bind to host cells. And in influenza on ancient Earth, it allowed it to jump between species. So the ability to move from swine to humans and birds to humans was caused by antigenic shift, or that reshuffling of the genome, resulting in our new virus. So the particular puzzle that I called you here today is because we have seen a new strain of Romulan cold breaking out across the quadrant. Originally on Romulus, you will find the dotted line would be your ID 50. So in this case, you have 10 to the 8th infectious particles required prior to half the population getting sick. But with this new strain, it seems to be closer. Our projections seem to have it closer to 10 to the 6th to 10 to the 7th infectious dose. What I need from you, cadet, is to determine what should we have the computer look for? What virulence factors should we examine that may explain this increase in ID 50 that we're seeing? At this point, we have no idea if this ID 50 increase will also show an LD 50 increase. And I need you to look into that as well. What should we have the computer look at to determine if we think this is going to be more lethal than previous strains? I thank you for your help, cadet. I'll send you back to the academy here.